So the measurement and the comparison of the total growth per unit time is called the absolute growth rate. Total growth per unit time is the absolute growth rate. Now the growth of the given system per unit time expressed on a common basis, example per unit initial parameter, is called the relative growth rate. Now first shows the maximum relative growth rate, then the second one. Okay, now condition for growth. Okay, oxygen required for growth, nutrients, micro required for growth, for the synthesis of protoplasm, and also excess source of energy. Now gravity and light affects the growth. Temperature also affects growth. Now differentiation, de-differentiation and redifferentiation. Now differentiation example is trachea elements. Now they at maturity, okay, the trachea elements lose their protoplasm so that they can carry the water through the large distance. Their cell wall has lignocellulosic secondary cell wall. Okay, it's elastic and it's very strong. Now you see the cells derived from the root apical and shoot apical meristem and cambium, that is secondary meristem, differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. Now de-differentiation, that means breaking of the differentiation. You see the living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. It kinds of rebirth. Okay, now this phenomena is de-differentiation. Example, formation of meristems, this interfascicular cambium, core cambium from fully differentiated parenchyma cells. So parenchyma cells, these are differentiated. Okay, now while doing so, such meristem tissues are able to divide. Now these meristems, interfascicular cambium and core cambium, they have the ability, okay, to divide again and produce cells that once again lose the capacity. Now these cells will divide and once again their product is going to lose the capacity. Okay, so this is redifferentiation. Okay, now example is woody dicotyledonous plant. Okay, tumor tissue culture. Cells position away from the root apical meristem differentiate as root cap cells, while those pushed to the periphery mature as epidermis. So root cap cells and epidermis, remember that. Okay, the growth in plants is open, that is it can be indeterminate or determinate. Differentiation in plants is open because cells arising out of the same meristem have different structures at maturity. So differentiation is also open. Okay, now development. All changes that an organism goes through during its life cycle, from germination of the seed to senescence, this is development. Now there is plasticity. Due to the environment or phases of life, plant shows different features. This is plasticity. Now example like heterophyll in cotton, coriander, larkspur. The leaves of the juvenile plant are different in shape from those in mature plants. So juvenile plants and mature plants, this kind of heterophyll in terms of like in terms of like what in terms of like what like in terms of like phases of life, like different phases of life, right? So when the differences in shapes of leaf produce in air, now this is due to heterophyll due to the environmental condition. So when it is produced in water, it has differentiate. So when it is producing terrestrial, it has differentiate. So example is buttercup, right? So the difference in shapes of leaves produced in air and those produced in water in buttercup also represents the heterophilous development due to the environment. So this phenomena of heterophily is an example of plasticity. First example, juvenile and adult. Second one is